Hi there. Welcome to our first topic for the Cambrian Explosion, where we're going to discuss what is known and what is debated about the origin of animals. So you'll hear more about the details of the Cambrian Explosion as we go through this subject, but basically it was a major diversification of animal groups from about 540 to about 510 million years ago. There was a big increase in the number of species living in the ocean, and it was also the appearance of many familiar types of marine animals, things that looked similar to animals alive today, including clams, marine snails, worms, shrimp-like things, and so forth. The Cambrian Explosion was also an important transition in ecology as more animals evolved to be able to move around, to burrow in the seafloor, and to eat each other. So just to put this in context and how it fits into Earth history, You've learned that bacteria have been around for about 3.5 billion years or more, but it wasn't until after the Great Oxidation event that more complex eukaryote cells evolved, perhaps 1.8 billion years ago or a bit more. But you still have to wait a long time, actually more than 1 billion years after eukaryotes had first appeared, for animals to evolve. So the first animals are a little over 500 million years ago. So if you wanted to relate that to, say, a 10-week quarter, animals would show up somewhere towards the end of week 9. So we're accelerating things in this class and getting to animals much sooner. So the Cambrian Explosion gets its name from the Cambrian time period of the geological timescale, which started about 540 million years ago. And fossils from the Cambrian time period, there are many of them that are clearly animals, that belong to groups that we know and are still around today. But when did animals first evolve? Did they evolve right around the beginning of the Cambrian Explosion? Or was that diversification event perhaps just an acceleration in a longer history of animal evolution that might go back tens or even hundreds of millions of years before the Cambrian even started? Well, we can first consider a group of organisms called the Ediacara biota. And these are large and entirely soft-bodied organisms that lived in the later parts of the Ediacaran period, from about a few million years before the Cambrian to about 40 million years before the Cambrian. And over time, they've been described as animals, or perhaps fungi, or perhaps giant protists, single-celled organisms, or perhaps even a completely extinct branch of life. So, with these, can we push back the history of animals before the Cambrian using these Ediacaran organisms? Well, first we need some terminology to talk about animal classification, and we'll do a very broad overview here. There are 3 main groups that often come up in these discussions. So there's metazoa, which is a scientific name for all animals. There's eumetazoa, which is the name for all animals other than sponges, or porifera is the scientific name there. And there's bilateria, which is all animals other than sponges and cnidarians. So we're ignoring a few groups here where the evolutionary relationships are currently debated, but this is the general picture. And we won't get into the subdivisions of bilateria here either. Another important concept is the distinction between a crown group and a stem group. So imagine we have 2 large groups of animals, say cnidarians and bilateria. Well, we can take all living cnidarians, jellyfish, sea anemones, and corals, and trace their evolutionary history back to a common ancestor, which is the gray dot here. We can do the same thing for bilaterians to get again the last common ancestor of all the living members of the group. And so everything that evolved from that last common ancestor, as it divided into multiple species and evolved over time, everything that evolved from it, whether those things are extinct or whether they're still alive today, belong to the crown group, in this case for bilateria, or the crown group for cnidaria. Well, we could trace the evolutionary history back even further to find the last common ancestor of all eumetazoans, so the last common ancestor of all cnidarians and bilaterians, which is the black circle at the bottom of the tree here. But you'll see that there are organisms shown by these branches that evolved after the last common ancestor of all eumetazoans, after the black dot, but before the last common ancestor of cnidarians, or bilaterians, the gray dots. And so these organisms belong to what we call stem groups. They're closer to one crown group, say Cnidaria, than they are to another crown group, but they evolved before the last common ancestor of crown group organisms. The yellow bubble here shows stem group bilaterians because they're closer to bilateria, they're on the branch to bilateria than they are to Cnidaria. Well, because life is arranged into these sort of smaller groups, nested within bigger groups, and so forth, things can be a stem group to one, so the yellow are stem group bilaterians, but they can be within a crown group of another. The things in the yellow bubble are also crown group eumetazoa. So this concept of a stem group organism is important because you might expect that some of these ancient organisms living many, many hundreds of millions of years ago might in fact be stem groups to the groups that are still alive today. But back to the Ediacara biota and whether they're animals or not. Well, one of the more curious features of many Ediacaran organisms is their unusual construction. They're made of these closed tubes that are arranged into kind of a quilt, like a pool air mattress, for example. In other Ediacarans, the organism is built in what we call a fractal, or a self-similar way, which means that a branch, for example, is made up of smaller branches, and each smaller branch looks like a small version of the main one. And those smaller branches have even smaller sub-branches that look like miniature versions again of the main branch, and so forth. These body plans, this fractal construction, and especially these closed tubes, are pretty unusual. They're not really like things that we find in animals, and so this is one of the reasons that some people interpreted Ediacarans as kind of a mysterious extinct group that's no longer around and didn't lead to any descendants later on. However, more recently, work has focused on growth patterns, especially how and where new pieces are added as these organisms grow in this segmented way. And the researchers here argue that these patterns of development aren't like those found in fungi or in other multicellular groups, but are more consistent with how animals grow, such as the fly shown in the picture here. So they felt that perhaps some Ediacarans could be stem group metazoans or stem group eumetazoans, while others perhaps could even be stem group bilaterians, and maybe some are actually crown group bilaterians or other groups. Well, a newer technique can now look at biomarkers from the fossils themselves. For example, here's a photo on the right of the classic Ediacaran fossil, Dickinsonla, and this one contains nearly entirely one particular type of biomarker, these molecules called cholesteroids with 27 carbon atoms, and these biomarkers are produced by animals, but biomarkers produced by fungi or other organisms were instead really rare in these fossils. But we might wonder, well, how do we know these biomarkers are from the organism and not just from the sediment itself? And how do we know they didn't seep into the rock later on, which is often a concern with chemical things? Well, the researchers actually measured the biomarkers in the fossil and in the surrounding sediment. And in the fossil, the animal biomarkers were, 8 or 10 or 15 times as abundant as the other biomarkers. These are the orange points in the graph, in graph D there. But when you measure the sediment just around the organism, there were more of the non-animal biomarkers, which are the gray points in the graph. So the biomarkers seeped in later on. You'd expect them to be pretty similar throughout and not different in the fossil compared to outside of the fossil. So the fact that only the fossil contains high abundance of these animal biomarkers seems to be pretty good evidence that Dickinsonla was some kind of animal. But what kind of animal and where it fits is still a little uncertain. Well, perhaps instead of looking at the fossils of the organisms themselves, we can look for traces of their activities. So trace fossils refer to trails or burrows that are left by the movement of animals, usually by bilaterians, but not always. So there is the presence of these organisms, the presence of trace fossils in rocks a bit older than 550 million years ago is often used to argue for bilaterians about 10 or 20 million years before the beginning of the Cambrian. However, as you've seen before, It is possible for simple trails or grooves to be produced by drifting pieces of microbial mats or other things as they get moved around by waves or current. So, how much does that reduce our confidence in these simple trails? Are they really made by bilaterians when we know they could be made in other ways? Well, one way to think about it is what's the most likely explanation and how much more likely is it than our alternatives? Yes, simple trails can be made in other ways, but bacteria and microbial mats have been present since at least 3.5 billion years ago. Well, trace fossils didn't really appear, except for a few controversial examples, until about 550 million years ago. So, yes, it's possible for trace fossils to be produced by drifting microbial mat fragments or something else. But then, if that's the case, why aren't there more of them from 1 billion years ago, or 2 billion years ago, or 3 billion years ago? The fact that they appear much later, in at least being common, suggests that something different than bacteria is making them, perhaps bilaterians. Okay, so the body and the trace fossil record likely extends animals or stem group animals in some way back to, say, 580 million years ago, about 40 million years before the Cambrian. But is there even older evidence out there? Well, let's look at biomarkers again, as we talked about with the evolution of eukaryotes and also with the evolution of cyanobacteria. Interest in this case is focused a lot on sponges because they're often thought to be sort of the earliest animal group to evolve for a variety of reasons. And there's a particular biomarker molecule. The name isn't important by a few care. It's called 24-isopropylcholestane. In any case, this molecule is found in sponges, but it's also been found in rocks as old as 650 million years ago. So maybe does that push the evolution of animals back, 100 million years before the Cambrian? Well, it turns out that the same molecule, the same biomarker is found in a group of algae. So maybe it's less certain. This is a common, sort of concern with biomarkers. Well, but this study took an additional step to test whether sponges or algae were responsible. And this figure is super complicated, so let's simplify it a lot. Okay, so the biomarker is found in rocks as old as 650 million years ago. So did it come from sponges or from algae? And both of them might have been around at that time. There were algae living then, and maybe there were sponges living then. However, it turns out that this biomarker requires a specific genetic change to happen for it to be made. So the scientists also used a molecular clock, which you've heard about before, to determine when that genetic change happened in sponges and when it happened in algae. And so they found that the genetic change happened in sponges, 600 million years ago or so, but that it only occurred in algae 400 million years ago, which was long after the biomarkers appeared in the rocks. So that seems to rule out algae as the producers of these biomarkers because they didn't have the genetic machinery then to do it. Well, however, another study later on, again found the same biomarker in a wide range of single-celled protists called Rhizaria, again sort of casting doubt on whether this is strong evidence for sponges 650 million years ago. All right, well, finally we can consider molecular clocks themselves. These have been used for around 30 years or so to estimate the timing of animal evolution. And there's been a lot of different studies, and different studies have somewhat different dates or different conclusions, slightly different. But in general, molecular clocks typically suggest somewhat earlier dates for animal evolution. For example, this study suggests that animals evolved nearly 800 million years ago, the blue dot branching point there. It also suggests that bilaterians, the orange branching point, evolved more than 650 million years ago. And so that would suggest, nearly a 200 or 250 million year long history of animals prior to the Cambrian explosion. And if that's the case, there are going to be really big gaps between when these groups potentially first evolved and when they actually appear in the fossil record. And so this has always been sort of a tension with the molecular clock versus fossil record studies. well, how can we reconcile, these differences and these results? Well, perhaps early animals were just like really tiny, and so they don't fossilize well, and we haven't found them. Maybe the molecular clocks are right, and animals were around for several 100 million years before we start finding fossils of them. Or perhaps the molecular clocks in some way like overestimate the age for reasons, maybe the groups evolved like in really quick succession, and the molecular clock spreads that out over time or something, right? So there could be reasons on both sides to explain this difference. So, what can we conclude overall from all this evidence? Well, animals very likely evolved before the Cambrian period, although the exact timing is still not that well understood. From the Ediacara fauna, it seems that there likely were animals 40 or 50 million years before the Cambrian period, but animals might have evolved, 100 or 200 million years or more before the beginning of the Cambrian.